Thank you, everybody, and welcome Duke alumni and friends to this session of the Duke Alumni Forever Learning Institute, an interdisciplinary educational program organized in a set of thematic courses. I am Bree Russell, Senior Director of Alumni and Regional Engagement for Duke University School of Medicine. Thank you for joining our program, Vanquishing Viruses. Today's program is a part of the theme, Fortifying the Body and Brain, inspired by the Duke Science and Technology Initiative. Moderating our discussion is the Dean of Duke University School of Medicine, Vice Chancellor for Health Affairs and Quadruple Dukey, Dr. Mary Klotman. Welcome, Dean Klotman, to the Forever Learning Institute.
Thank you, Bree. I'm so excited to be joining all of our fellow alums. And truth be told, I am doing this from New York because I'm going to the Duke basketball tournament tonight. So very exciting in both hosting this and going to the tournament. Today, we're going to touch on that expertise that we have in vaccine development. This is an area that we have shown very strong progress, and I think you'll all be proud as I'm the university's global leadership in vaccine research. The title of this series certainly applies to the topic of forever and continuously learning. As we've all been reminded in these pandemic times, there are always newly emerging infectious diseases or the reemergence of old infectious diseases, sometimes coming back to the development of drug resistance or the ability to evade former vaccines. So the work is never really done. Vaccines, which train our bodies to recognize a pathogen before we become infected, certainly matter at the individual level. But what we have certainly been taught during the pandemic is these are really important tools for public health. And because infectious diseases often have a bigger impact on the economically disadvantaged, vaccines are a force for social equity and justice and for simply making the world better. So we're so proud that Duke is at the forefront of vaccine research, and much of this exciting work is being done in our own Duke Human Vaccine Institute. Formed in 1990, DHVI's core mission is to research, develop, and test innovative diagnostics, vaccines, and therapeutics to prevent and treat global diseases. The DHVI team pursues new avenues for disease prevention and treatment and translate those discoveries into vaccines for diseases, both new and old. Because DHVI was formed in the height of the AIDS epidemic, a key focus of our work has always been the search for an effective HIV vaccine. The goal is to develop a vaccine that will eliminate HIV from the planet, and we're getting close with the tools, the knowledge, and the plans in place to create it. But this has been a daunting task. And many of the lessons we've learned and techniques developed during our work on HIV vaccine have informed our contributions to the development of a COVID-19 vaccine. In fact, the prior fundamental work, infrastructure, and technology allowed us to rapidly focus our efforts around the question, could we develop one vaccine that protected patients not just against COVID, but against all potentially dangerous coronaviruses and variants? And we're getting close. Duke researchers recently developed and demonstrated proof of concept of the world's first pan-coronavirus vaccine, now preparing to move into clinical trials. This is an enormously exciting development, and I'm sure we'll talk more about that today. At the same time, we're also working to improve influenza vaccines. The home run would be to develop a universal flu vaccine that offers permanent and multi-year protection against multiple strains of influenza, including those that have not yet emerged. Between now and the home run would be smaller, meaningful gains, including better protection of older adults, longer lasting protection, and faster manufacturing processes. Speaking of manufacturing, many people don't know that DHVI also has a production program that enables us to move vaccines and therapeutics from basic science discovery to clinical trials. Our facilities are small compared to commercial plants, but they enable us to make the products we need to follow the science and rapidly test the proof of concept. Our infrastructure also includes a regional biocontainment lab that we can safely work with the most dangerous pathogens or new pathogens where the risk is not yet known. This allowed us to work with SARS-CoV-2 from the beginning. And we're home to a repository of human blood and tissue samples that enable research with institutions around the world. So it's fairly unique to have research, development, manufacturing, and clinical teams all working together. And it is very effective, allowing us to be nimble as we move ideas forward. Developing experimental vaccines can be a laborious process of creating, testing, modifying one formulation from another. So having our own manufacturing capability in-house speeds up our timeline and makes us more efficient. And last but not least, DHVI is also has an educational component. We're one of 8 sites nationally to serve as a hub for training first responders and other workers on how to safely deal with infectious diseases that are emerging, like Ebola, Zika, malaria. So what makes us such a strong force for vaccine innovation? It's simply team science. You may remember from the time at Duke that this is a highly collaborative environment where teams tend to form across boundaries, bringing together science experts and working together across disciplines. And we're building that approach into our brick and mortar facilities as well. We have a major new hub in the Research Triangle Park called Duke at RTP, which is a home to scientists from across Duke for vaccine development. That environment creates a cross-pollination of ideas, interactions that can lead to innovation. So that's a little bit about DHVI. And while I have the floor, I want to spend just a quick moment to say why it's so important for all of us to be ambassadors for vaccines. In a season when we're experiencing a tridemic or more of RSV, influenza, COVID, and other respiratory viruses, the best thing we can do for vulnerable populations and for society as a whole is to get vaccinated and encourage others to. COVID vaccines alone prevented over 20 million deaths a year, and yet more could have been prevented were it not for society's vaccine hesitancy. So I would ask all Duke graduates to join me in standing up for science and reminding everyone that vaccines work. And if you also feel moved to help fund our research and technology, all the better. Science is expensive, but it's the best way we have to guard against emerging diseases and ensure a better and healthier future. So now to our discussion. Here with me today, I have Dr. Georgia Tomaras, who's professor of surgery and recently announced chief of the Division of Surgical Sciences in the department, as well as professor of immunology, molecular genetics, and microbiology. And we have Dr. Kevin Saunders, associate professor in surgery and molecular genetics and assistant professor in immunology. They are both joining us now to answer some of the critical questions. So with that, let me start asking you both a question as to what motivates you to do the work that you do. So Kevin, let's start with you.
All right. So I've always really been fascinated by science. And so really early in life, I really gravitated towards that. And what I wanted to do is actually have a career that could help, human health. So I wanted to have an impact on people's lives. And I never really had the drive to want to treat patients, in that setting. But I found that science was a way that I thought I could do that. And so I really wanted to have a translational science career where the things I did in the lab could actually have real life implications. And that really drove me to studying viruses. And I did so here at Duke. And that's where I really fell in love with really trying to understand how does the immune system fight off viruses. And that work really kind of transitioned over from just understanding how our immune system works and how it fights viruses over to can we make vaccines that can actually have that kind of impact and actually protect us from viral infections. And so it's been a kind of a progression from just understanding the basic science behind how viruses work all the way up to more translational work now where we can make vaccines that can go into clinical trials.
And Georgia, what about you?
Yeah, so my research is really, my lab are really driven by the desire to understand the features of a protective immune response. As Kevin said, it's really important to understand the immune system and how we can resolve disease. So our lab is really focused on studying antibodies that can protect against COVID infection, influenza, HIV. And our research really helps to understand how we can teach the immune response to effectively adapt to prevent disease. And then we can utilize that information to directly contribute to the design of clinical trials to have those direct interventions to improve human health.
Now, neither of you were working on coronaviruses before the pandemic. So I'd love you to talk a little bit about how you were able to rapidly change directions. Kevin?
Sure, Dean Klotman. So when the pandemic first started, we didn't really understand how large this would be. And so we had this toolkit that we had developed for HIV research and HIV vaccines, which, as many people know, it's one of the more difficult vaccines to try to develop. And so we had this extensive toolkit, and we were watching as this virus kind of spread not only across the world, but across the United States. And we thought that, hey, we really have the tools that we would need to jump in and have an impact here. And we had a facility here that could make a clinical product if we could come up with one. And so the first thing we did is we said, well, let's try to understand what are the most effective antibodies that one could make that could prevent infection. And so we started to study just the basic science and basic immunology of how people were responding to COVID-19 infection. This was actually particularly difficult given that there weren't a lot of local cases of COVID-19. And so we were kind of waiting in the wings, waiting for samples to be available. And luckily, there was this cohort called the MESI cohort that came about by Duke investigators that were going out and collecting samples. And that really enabled us to really try to, early on study how COVID-19 was triggering an immune response, and then what that immune response might be that we'd want to also trigger with a vaccine. And so we paired together our work with antibodies and being able to find the best antibodies that can prevent infection. And then we used those antibodies to design a vaccine that could generate that same type of antibody response. And this was all a model that we used with HIV. So ultimately, we basically took our toolkit that we already had and just applied it to the COVID-19 pandemic.
Yeah, when people talk about it, it's amazing how fast the vaccine emerged. It's actually based on many, many years of research and having the tools. And I think that is something that we have to keep building on. So Georgia, what about you? How were you able to change directions?
Yeah, so like everyone, our work was really immediately impacted in how we were proceeding. We have our global health partners that immediately reached out to us and asked us how we can utilize the best test to measure antibody responses in human clinical trials, how we could adapt existing technologies that we've developed, and how we can learn about the biology of SARS-CoV-2 to adapt our systems to analyze the responses. So our team really pivoted quickly, that we learned as much as we could about SARS, learning from past literature, and had conversations with experts all over the world. So there were a lot of virtual calls with people who had been working on SARS previously, and then interacting with other labs that are working on it and newly developing new technologies to pivot their labs. So we made immediate investments really in people, scientists who had the right expertise and develop new technologies so we could better evaluate the immune responses. As part of this too, we had to really look at a research portfolio and decide on which studies could wait and which ones we still needed to move on going forward. And, one of the ones that we did in parallel with COVID was an HIV clinical trial that was very high profile, looking at prevention. And our dedicated scientists at Duke were really willing to keep going. They had to work through the night, and over the weekends we had to rotate our research program so we had enough space for everybody, had enough protective gear. But we're really grateful to them to apply their expertise during this time to keep going. And then they were also able to pivot their technology to looking at SARS-CoV-2 infection. And then in a sense, this work has really benefited our other programs. So the technologies and the way we work for SARS-CoV-2 and for COVID vaccines has positively, I'd say, impacted how we're working for HIV vaccines and for influenza.
Yeah, it's hard to remember what was happening those first couple of months, but to your point, we had to make it a safe environment for investigators, and they came back to work and everybody changed protocols, but it was just extraordinary how we were able to do that, and largely to the credit and passion of our faculty. So Georgia, vaccines obviously have been around for some time, but the innovation the last 10 years, I think, has been extraordinary. Can you talk a little bit about some of that really helped move this forward as rapidly as it did?
Sure. So yeah, and I can speak as I'm one of the PIs for the HIV Vaccine Trials Network. That's an international organization to test vaccines, some of which Kevin is producing, and we're now able to test in the clinical trials. But I think in the last 10 years, the field, it's actually really remarkable how much it's changed. Probably four main ways. The first is that many investigators are now looking to have more specific and focused vaccines that can better influence the immunogenetics of the host to drive, in particular, particularly difficult to elicit antibodies, what we call broadly neutralizing antibodies, but a deeper understanding of immunology, also how different immune cells talk to each other, how they chat, how they communicate. When you see the pathogen or HIV in any form, how is that presented effectively by maybe a dendritic cell? How does that effectively lead to a better T cell and B cell response? And then how to leverage the biology of what we understand and what happens in the germinal center, where much of the action happens for how we educate an antibody. Our immune system educates an antibody to be protective against a pathogen. All of that biology, there's so much to uncover. And I say the field has come together to really try to understand that complexity so we can design that in the immunogens. At the same time, I mentioned that the immune system is really an interactive network, of cells and molecules that work together to protect our body. So really understanding that coordination and that synergy of responses has really come a long way, especially with a lot of new computational tools, new methods to understand the immune response, and then pulling them together to fully understand them. And then I think the last way is one that Kevin is really closely familiar with, is immunogen design has changed dramatically related to understanding the structure of the regions on the HIV that are most vulnerable, and in particular, leveraging some mRNA technologies. When we look at our long list of vaccines that we have to test, and again, for the HIV Vaccine Trials Network, it's a long list of things that includes mRNA, includes fine structures that have been put together by scientists like Kevin.
Georgia, you might explain a little bit what an immunogen is.
Yeah. So, and Kevin, you should take this one. This was punted to you. I think an immunogen we see as something that's stimulating the immune system, right? So it's being presented in your body in a way that can stimulate your immune response, hopefully in a protective manner. But the immunogen has fine designs depending on the pathogen of interest.
So, Kevin, everybody knows mRNA vaccine today,
Right.
but it actually didn't exist, at least in the clinic, before COVID. Can you talk a little bit about how that is so different from our more, traditional vaccines?
Right. So probably traditionally, we're more familiar with inactivated versions of the pathogen. So meaning that you heat it up so that it's no longer infectious, or you add some chemical to it that inactivates it. And, you would deliver that to someone, they generate an immune response, and some part of that immune response would be protective. And, a lot of times we weren't really sure exactly which parts of the immune system were actually protecting, which types of cells were mediating that protection, but you just knew it worked. And over the years, we've kind of shifted away from that approach to something that's more fine, so more detailed, more rational in a sense, where you really want to focus the immune response to the part of the pathogen that's going to give you the protective immune response. And that's really been done with subunit vaccines. So that means you take a small piece of the pathogen, the part that you want the immune system to see and focus on, and you deliver that. The body then sees it, it then responds to it, generates an immune response to it, such that if you do come in contact with the real pathogen, you already have an immune response that's trained and ready to respond. And that's really where the protein-based vaccines kind of shifted to. And it was viewed that those would be impactful, but also be perhaps more safe. And the next generation of vaccines have really shifted to being able to deliver the genetic information for that small piece of the pathogen. And so that's where the mRNA vaccines really took a hold. And it's not that people hadn't tried that before. They tried it with other genetic information like DNA. But the mRNAs really had the right technology that came together to make them really effective. And so you could put together precise designs where you were only going to show the immune system the part that you wanted it to focus on. And usually that's the protein that's on the surface of your pathogen that you want the immune system to see it, even before it's able to infect cells and hopefully prevent infection of the target cells. And if you combine that with something like mRNA, where you can deliver that information, have a great robust response to that part of the pathogen, then you have a great chance of being able to make a protective vaccine. It also gives you the advantage of being able to make the vaccines a lot faster. So in the opening, we talked about, making the next generation of influenza vaccines, and that, having processes that can move forward a lot faster. Something that's not as laborious, not as prone to perhaps, production issues and problems and challenges. If you can do something like make an mRNA that's fairly straightforward to do, It gives you a lot more flexibility in how quickly you can make vaccines, how broad they can be. You can make them against different parts of the pathogen, different pathogens from different areas. The flexibility of what you can do now is really opened up with the technology that's out there.
So that kind of begs the question, are you incorporating mRNA technology into your vaccine work?
Oh, definitely. So we've published, I think now, something like 3 papers now that all have mRNAs as part of the main vaccine that we've used. We're using it on both our HIV vaccine program. We did some initial studies first to just compare how mRNA vaccines performed versus protein-based vaccines. And we found there that the mRNA vaccine was as good or better in most of the cases where we compared them. We then shifted that over to a lot of our COVID-19 research and are using mRNAs as a way to deliver the antigens that we design as well. And so, yes, we have ongoing studies in both the preclinical side, but also the clinical side. So we have clinical trials that are scheduled for early next year and on through next year through 2024 that will deliver mRNA as part of our HIV pipeline.
So before we move to the next topic, I'm going to ask both of you this question. What's the downside of mRNA vaccine technology?
Is Georgia going first?
Go ahead, Kevin. Yeah.
So downsides. So one of the challenges with mRNA is being able to appropriately express the antigen or the immunogen that you selected. One of the advantages of protein is that you can make it in a factory somewhere. You can quality control it. You know exactly what it looks like. You know how it folds. You know all the ins and outs of the chemistry behind that particular part of the vaccine. When you deliver that same genetic information for that same immunogen via mRNA, you get whatever the body makes. So it can make some of that looks exactly like what you want it to look like. So it makes the protein from the SARS-CoV-2 virion that looks exactly like what's on the virion. But it can also make misfolded, unfolded versions of that same protein that maybe are not helpful. And so there's a balance there where you have to have a really nice immunogen design that gives you the type of immunogen that looks like what's on the virus. And, that's where the structural biology comes in. That's where the biochemistry comes in to really designing a vaccine that is effective. You can't just take any protein and turn it into an mRNA and then it just works. In some of our HIV work, we spent somewhere close to 18 to 24 months just iteratively making new versions of the vaccine immunogen until we finally found something that we could express by an mRNA. And so there's some work that has to be done when it comes to really giving the best version of your vaccine if you're going to deliver it as an mRNA.
Right.
And I can add that,
Right.
in just thinking about the immune responses to mRNA versus protein, as Kevin mentioned, there's a lot of side-by-side comparisons where mRNA is doing as good or better than other traditional vaccines. There's some things that we still don't understand about the durability of that response. And as Kevin said, differences in the structure of what's made from an mRNA versus a protein. And then I think one of the overarching themes from this too is we probably need diversity in the types of immunogens that we have in the field. And so not a downside, but mRNA could be just one additional tool to the different types of vaccines that we have. And we need many different types of vaccines and how they look, mRNA, protein, or other genetic material. So we can then better understand how to get a better protective immune response. And in some cases, like we've seen in COVID, right, with many vaccines that work, like very different vaccines that work. And this leads to choice for populations in which vaccine they can easily manufacture and bring to their own populations.
Well, Georgia, I don't know if you're reading the chat, but I'm going to introduce one of the questions because it's very relevant to this conversation. So Corbevax, which is, a very inexpensive vaccine developed out of Texas Children's and Baylor College of Medicine, is now being used broadly around the world, particularly in India. What do you see if you're comparing that approach versus mRNA? What are some of the important differences?
Yeah, so again, I think the comparison of they're eliciting similar immune responses across vaccine regimens. The data that's been out there for the public is a first look at circulating responses for the most part, looking at antibodies that can directly neutralize SARS-CoV-2. So they bind to the spike and prevent it from attaching to the host cell, and binding antibodies, just looking at the specificity. So we know that many of these vaccines can achieve protective levels of those antibody responses. There are some differences, however, among vaccines in the total immune response that they elicit. So the full breadth of that, the specificity of those antibodies, like the multiple targets, as well as its coordination with the T cell response.
Kevin, thoughts there? Just thinking about the Corbevax versus mRNA approach.
Right. So I agree with Georgia. So there's really, if you go back and look at how many different vaccines are out there, I mean, I think when the pandemic first started, there was estimated something like 150 different companies that all had vaccines entering into phase 1 clinical trials. So I think there is more than one way to get there. I think overall, the idea is that if you have something that can be perhaps a little bit more specific in that it's using just a single antigen or a single part of the virus, that perhaps that vaccine would be more specific and generate immune responses that would be more tailored to the protective types of antibodies that you want to generate. However, with mRNA, it does seem to be still somewhat debated that it's not generating the types of other cellular immunity that you might would want. And so there may be a place for something like the more traditional types of vaccines. I think there's going to be a place for protein-based vaccines eventually, given just the durability of the immune response, that, you get this great immune response that kind of comes up, peaks, and then seems to kind of wane over time. So I think some of the more traditional vaccines will be able to fill that space and hopefully come in and generate a more durable immune response. And I don't think of it so much as either or. I think that they can hopefully work together, given that many of us will have already received an mRNA vaccine. Knowing how the more traditional vaccines work as a boost, and how that immune response that was generated by the mRNA vaccine can hopefully be augmented and hopefully become more durable using a more traditional vaccine is the way I view it.
Can I add one thing to it? Because I think the chat, like that question is so rich with other comments too, in terms of being able to manufacture in country too, right? And no patent and being cheap. And I want to emphasize that this is very much in the discussion, right, with us that are working on vaccines, of how do we make it equitable? How do you get vaccines everywhere? Good side that we've talked about, that, regardless of the vaccine type, at least for COVID, as long as you're getting a certain immune response, you're getting some level of protection. But everybody's thinking about how to do this, how to make sure that we're getting manufacturing in country. And one of the things that is really critical for the work that we're doing is understanding the immune responses that track with protection. And what we call that is a correlate of protection. And if you can identify correlates of protection, so this could be one response, right? Or it could be many. But if those are consistent for the vaccines, no matter what you use, protein, mRNA, no matter if it's manufactured in the US or India, that's a really important marker that you can then bridge any vaccine regimen such that any country can utilize their own technology. And as long as they get that immune response, then they know that they're going to see protection. So that is, a major area that we've been working on, because that will enable all manufacturers to actually produce vaccines that work.
That's a great point. If you know what the indicator of success is, then, you could get there probably a number of different ways. I'm looking at the chat, and there's such great questions relevant to this conversation. So I do want to follow this a little bit, because I think a more general question is related to specifically mRNA, but with vaccines in general. Why do you see such a waning of antibody response? It's not unique to mRNA, but why is that? Kevin, you want to start?
Yeah, so I think there's two schools of thought. So one of the schools of thought is that it's something specific to SARS-CoV-2, that the spike antigen itself, that's what's included in the mRNA vaccines, the portion of the virus that's been selected to be encoded by the mRNA vaccine, that the spike protein itself will generate responses that are not so long-lived. And perhaps that's because you've been exposed to other coronaviruses throughout life. And what you're basically bringing forward are some of the antibodies that cross-react with pre-existing or with the common cold viruses that you've seen in the past. And so if some of the responses are due to that, and those aren't the types of B cells that tend to hang around for a long time, then your antibodies wouldn't hang around for a long time. And so I think some of that is part of the idea. The other parts of this is that a lot of times it's thought that mRNAs are not generating durable responses. But in the preclinical studies that we've done, we found that mRNAs can give great durable responses. We did a study using Zika virus as a model system where we delivered Zika virus proteins using mRNAs, and the animals generated neutralizing antibodies that were at a high enough level to protect those animals from infection. And those antibodies persisted for over 42 weeks. And so they never came down. Once the level was established, they stayed there. And so it at least suggests that mRNAs can do it. And maybe it's not just that mRNAs are somehow flawed in their ability to generate a durable response, but it's something specific to the SARS-CoV-2 protein in particular.
So, Georgia, a related question is, what is the role of memory cells? And, we focus on this waning neutralizing antibody response, but if you have a robust memory population, can that rapidly be deployed?
Yeah. And that's a great part of this question because right, you want a certain level of antibody that's pre-existing to prevent infection. But then even if you have waning or you have a lower response, as you said, your memory recall will come into play, and you will soon have enough antibodies that are present to help ward off disease. So we're seeing that, I think, with our COVID vaccines, or many people have been vaccinated and boosted and they're still getting infected. Some of that is you already have prior immunity and there is memory to SARS-CoV-2. So you are preventing severe disease. You may not have prevented acquisition, but you are preventing disease. So that is the good part of, even though it's waning, you have a protective response. And I think it highlights an area that we've been really excited about now in looking at is the role of IgA in the nose in terms of if you can elicit a robust local immune response, in this case, because COVID is a respiratory virus, and it's coming in through the nose and the respiratory tract. If you have pre-existing antibody responses of a certain form, which is IgA in abundance, can you actually better prevent acquisition in addition to preventing disease?
So I think it's really fascinating that all of a sudden people are focusing on the nasal mucosa, which is the initial site of contact that we haven't thought much about, but it's obviously incredibly important. So, both of you, I want to talk a little bit about team science, because my introduction, I said that vaccine development is really team science. So, Georgia, we talk about One Duke. What does that mean to you in terms of team science?
I love this question. I love it in talking about the immune system too, because we're talking about how parts of the immune system work together as a whole to lead to protection, right? And one of the things that we're studying is trying to understand infections with people that do really well at preventing infection or disease, and how do we understand that to how to replicate that in a vaccine. And for this, like One Duke really is exemplified by the research units across Duke for this, because our scientific community and culture and the intersection of basic clinical and quantitative sciences really helps us work and solve this problem, which is essentially One Duke. So in terms of research units at Duke, we really do work across departments, different disciplines, institutes, and centers. In particular, the institutes and centers I'm a part of are really comprised of multiple investigators coming from different disciplines with different expertise and training. So that really enables the work. I also, to get this kind of work done, where we're counting on so many different types of expertise, that the environment and culture is really important. So I think, in general, people are seeking out embracing different viewpoints, because that really helps us understand where we need to move our science and look at things that we've not looked at before. We also recognize that we're all learning this together, right? We're recognizing that everyone has something to contribute. And many times it's just a question, like what we're getting in the chat from people. The questions that you ask really inspire us to think about a problem in a new way, turn it over, and to be able to learn something about it and maybe influence the direction of our work. And then finally, really the intersection of One Duke across, basic clinical and quantitative sciences. Our lab is really a part of a clinical department, basic science departments, a center and institute, a center focused on quantitative immunology. And that's really a key now that we have. We're gaining more understanding of the immune response. we really need computational power to pull this information together so we can cohesively understand the immune response. And that's going to take multiple types of people working in different areas at Duke to achieve this going forward.
Yeah, I mean, the need and power of data science behind this work is just extraordinary. And that's something that I would say has really been the last couple of years, because our ability to generate data sets is unlimited, but we really need the data analytics to be able to make sense of it. And so that piece has grown at a very rapid pace. So Kevin, let's say you've got a new pathogen and you want to really start thinking about how to develop the vaccine. What is your approach, in terms of putting the people together and really starting, the work in something that you haven't done before?
Sure. So the first thing you'll need safety. So we have a team of safety officers here that make sure that whatever it is that we do it safely. And so that usually leads to making sure you've got the right lab space that has the right containment, so that anything that's scary doesn't make its way out. So there's no threat to the outer public if we're working with something that's new. The next thing we usually need is virologists. And so these are the folks who can make more of the virus itself, so that we can then study it. We can study what immune responses can block it from being able to infect cells. Sometimes we don't even know what cells it can infect. And so we kind of start just with a panel of things and look, where exactly does the virus go and where does it end up infecting? And then usually where my team kind of slides into this is then we look at the immune response. And where we go is normally to people who have become infected, but have been able to clear infection or have somehow otherwise managed the infection. And those are usually great candidates because they can show you what the immune system needs to do right. And if you can then generate that type of immune response, either with a drug or a biologic or a vaccine, then you have a great chance of having that type of, helpful immune response show up in all the people who are at risk. And so our team jumps in and starts to study what are the types of antibodies that are made. We know that antibodies can prevent infection, so that's usually where we start. We use all the tools that we have available to be able to understand all the way down to the atomic level of what the antibody does. We then have a team of structural biologists that then start to work on designing a vaccine. And so we take the information from how the antibodies interact with the virus. We then trim down the parts of the virus to focus in on just those parts that the antibodies are recognizing that we think are critical. And typically what we do is we make what we call a nanoparticle vaccine. What the nanoparticle vaccine is if you could picture kind of like a soccer ball where you have these different little hexagons that come together to form a larger sphere. And each of those hexagons have fixed to it a part of the virus. And this forms this ball that looks kind of like a virus, but doesn't have any ability to infect cells or have any of those kind of safety concerns. But when your immune system sees it, they see this ball that's kind of floating around that looks very much like a virus. And that triggers your immune system to really kick in and to make antibodies against that critical part that you've displayed. And so we use that mostly as our platform, and that then generates, really high antibody responses against that critical target. That's where we start to move forward with trying to understand how well we can protect against infection. And so it's a full pipeline of just understanding from the very basics of, where does the virus go and what cells can it infect, to what's the Achilles heel on the virus so that we understand what we need to generate immunity to. And then we have really cool structural biology techniques to, to basically focus the immune system on those critical sites.
I'd also add that at some point early on, you have enough data, you start thinking about who will fund the work, which is really, really important.
That is key.
That is key.
As you and I have discussed before, the hardest part, the hardest part in responding to a pandemic is that you don't have funding to actually do that. Your funding has been directed to other pathogens. And so you may be well funded and have all the tools that you need, but none of that money has been allocated for this new pathogen. And so really you have to become, somewhat more flexible in understanding, how do you get funding for that? What are the sources that have immediate funding that you can go after? And sometimes funding comes from places that we don't expect. Sometimes it's donors that have seen a pandemic and want to try to jump in and try to help with that pandemic. This past, for the COVID-19 pandemic, we actually received funding from the state of North Carolina. And that was somewhat of
first time ever. First
Right.
time ever.
So sometimes it comes from places you don't
Yeah.
expect, or you don't think will actually be part of the solution. But without that money, our vaccine would have never been made. We wouldn't have been able to really have an impact on the COVID-19 pandemic without that funding. It was absolutely essential to the work we did.
Yeah, I mean, to Kevin's point, early on, we had a conversation and he kind of said, Dean Klotman, don't you have money for this stuff? I'm like, no. We were very fortunate that early on we did have donors come forward and give us some flexible dollars to support the extraordinary work that a lot of our investigators wanted to do. But at one point, at some point, we reached a level of need and expense that we really had to look externally. And North Carolina did come through in a very extraordinary support of a private institution, which was great. But it exposed an area of concern that we need to work on going forward is having the ability to support research in a way that we didn't plan, and thinking about what those sources are. So yes, we did have that conversation. Georgia,
Yeah.
what do you think we're really as an institution poised to make a big difference in vaccine world? What do you think is the Duke special aspect?
Yeah, and I think we touched on this a bit before too, is this idea of coordination of the immune response and looking at local immunity. Those are probably two areas that we could really push forward. Our lab had discovered a role for specific antibodies, these forms called IgA and IgG3, that were important for mediating differential effects for protective HIV vaccines. And we continue to identify the features of antibodies that are important for protection, such as the form of antibody, how they target the pathogen, the strength of that pathogen. And we've also recently found that, in looking comprehensively and using computational tools, that we can identify immune profiles of groups of individuals that respond in one way or another to a vaccine or to an infection. And we really need to explore that more. We did this with COVID and looking at people who had, were able to contain virus shedding in the nose, for example, had a shorter period of time of shedding versus a longer period of time. And so as we think about the immune response, and we know that there's a coordination and there's multiple layers of immunity. So we have to think of this as protective layers, that if one fails, we have another layer underneath it. And at the same time, the immune system is working together kind of like a band or an orchestra where everybody has a role to play. And then the final output is a beautiful sound. And this would be, in this case, like protection against a pathogen. And two areas that we really, I think, at Duke are poised to play a role is the intersection of our clinical and quantitative sciences, and understanding that local immune response and pulling that together in a comprehensive manner to understand what constitutes protective immunity and how do we understand that in different people. I saw another question in the chat about variation in immune responses, and we've always been struck by that, right? Everybody gets the same vaccine regimen and you profile their immune response and they're different among people, even though everybody's getting the same vaccine. So we know that your prior exposure to other pathogens, the environment, to other vaccines may influence your responsiveness. We still don't fully understand how to sort of modify that variation and say, if we know what a protective immune response is, if we have those immune correlates, now how do we get them up in everybody to that protective level? And I think understanding the variation among all of us, why are we so diverse in our immune response? If we can really understand what drives that, we can probably then leverage that in a vaccine and say, okay, some people who might have a lower response or weaker, let's bring them up specifically. Let's understand what that is and either provide them a different strategy or more of the same strategy, whatever it takes to bring everybody up. But we're still at a phase of really understanding what's that underlying biology for that difference in variation.
A related question to you're talking about understanding the role of the mucosa is one of our participants asked, are we working on a nasal vaccine? Either Kevin or Georgia.
Yeah, Kevin, go ahead.
Yeah, so we directly are not working on a nasal vaccine, although we have partnered with some groups that are doing that. So there's biomedical engineers that have come up with a system of chemicals that you can mix with your protein of interest that then basically takes it and allows it to be slowly released at the site that you care about. There's also a group that's working on mRNA delivery intranasally. And so I know that there's some work that's being done on that will, at some point, some of our immunogens may make it into those types of studies as well. But as far as, like physically our labs here at DHBI, right now we're more collaborators on those types of projects.
And then on the other side, so our lab looks at the immune responses to vaccines, so systemic or a nasal vaccine. And we also have partnerships looking at that. And we're really trying to explore what are the immune response that are occurring there. So again, looking at IgA, being able to build those assays so they're very specific and sensitive and can be compared across regimens and also transferable. So as other labs are looking at that, and if we have a marker of IgA that's a protective immune response, we can immediately transfer that method to another group. So there's some technical work that we're doing here at Duke. And then also like method development, novel discovery of saying, we know antibodies are present. There are other cell types that may be present that are kind of elusive in some of these mucosal secretions, and developing new methods and new technologies for studying these immune cells are going to be really critical for understanding the biology. So when Kevin and others make these vaccines, we can say, oh, okay, this is the immune response that's being generated, and it is or is not being protective.
Right. And in addition to that, Dean Klotman, so vaccines is one topic, but we've also worked with groups who are actually taking the genetic information for antibodies. So if you have an antibody that is protective, they can take that genetic information, again, formulated with their special chemicals that can deliver it to the nose, and then show that when they do that, the antibody can be expressed and actually is protective, at least in the animal models that they used. So it's not just a technology that we're using for vaccines per se, but also for just the delivery of protective molecules.
Right. So Kevin, I talked a little bit in the introduction on the pan-coronavirus vaccine. Can you talk a little bit about what that is and where we are in testing the efficacy?
Sure. So when we initially started to work on a vaccine, we didn't think that we'd be the first ones to make a COVID-19 vaccine, since we knew from the partnerships between Moderna and the Vaccine Research Center that they were flying forward. And some of the meetings that we had with them on HIV, they were talking about the progress they had made with coronavirus vaccines. So we knew going into it, we weren't going to be the first ones to make a vaccine. So our goal all along has been, can we make something that could come in afterwards that maybe could broaden protection or even boost protection higher than what the mRNA vaccines were doing for COVID-19. The way that we did this was, again, using our antibodies to guide us. And the antibodies that we found really guided us to the receptor binding domain of the SARS-CoV-2 virus. And this is a portion of the virus that attaches to the receptors on cells that it wants to infect. And so we knew that if we could generate antibodies against that particular part, then we would have the ability to protect against infection. And so what we did is we took that portion of the virus and we stuck that on the surface of our nanoparticle. This again generates something that looks similar to a virus, but has only the critical part that we want the immune system to see. That generates a really robust response against that portion of the virus. And those antibodies, much to our surprise, not only were great at protecting against SARS-CoV-2, but could also protect against many other coronaviruses that were similar to SARS-CoV-2. And these included viruses that circulated in bats. We think that's important because we think the next pandemic that would have this kind of same potential as SARS-CoV-2 will probably come from the coronaviruses that circulate in bats. And so that's been kind of the general platform that we've used. Since that initial molecule that's gone forward now and is in manufacturing here at the Duke Human Vaccine Institute, we've also done the same approach for other parts of the spike protein that we know are critical for the virus to be able to infect cells. And so there's two other sites that are more, closer to the surface of the virus that are also conserved and can also be targeted by antibodies and also can give you this broad protection if you're able to elicit those. And so ultimately, I think the pan-coronavirus vaccine is going to be one that targets the receptor binding domain, but then also these two conserved sites that are more in the stalk region of the virus.
So, I mean, both of you have answered a question that I was going to pose to you, which is, why do you think Duke gets these enormous grants like the flu, the big flu grants, or HIV? But I know the answer. You know how to do team science. You collaborate where it's needed. You have an amazing group of investigators, and you're adaptable. And so I know the answer to that one. I love some of the questions coming up, so I'm going to continue to check the chat. But one that's particularly exciting is, how does this work potentially overlap into areas like cancer? And can we imagine vaccines to cancer? And I love that question because I think that is our vision. But Georgia, do you want to take that one?
Yeah, sure. And I think this gets at understanding the immune response in each individual and targeting that specifically, because that's ultimately where some of the cancer vaccine strategies are going. You have to understand the disease state first, and then make the immune response specific for that disease state. And that can vary between individuals. And the technologies are getting better to assess that. What are the targets? And then how do we amplify the immune response in those individuals specific just for the tumor? And it is similar to a vaccine problem. You have to know your target, you have to know what you're going after, and then you have to fine tune your immune response to make sure that you're eliminating either that pathogen or a tumor formation. And I think then it gets at this idea of variation among people and why we have different responses, so that we know there's different responses to therapeutics as well. If we can understand differences in vaccine responsiveness, we can also figure out, is it similar? Are those features also varying how people respond to a therapeutic intervention?
I would argue that a lot of the technology we're developing, for specifically pathogen, infectious diseases, can be used for harnessing an immune response to a cancer or other areas. So I think it's a platform that we at least are thinking about a much broader application. But Kevin, are you thinking about application to cancer at this point?
So it's funny because we've watched the cancer vaccines and cancer treatments and actually applied that to the vaccines that we're making for infectious diseases. So I think there is a lot of crosstalk between the two.
Right.
In particular for cancer, they're really trying to boost the T cell response that can go in and clear the tumor cells. And, having a great T cell response against a pathogen never hurts. And so being able to understand how you can modulate it in cancer also informs us on how we can modulate it for HIV or for some other pathogens where cells can become infected and we need to clear those cells prior to really having someone say that they're cured from from a particular infection. So I think being able to modulate T cells is one of the key areas that I think kind of overlaps between the two. And so the basic immunology that really underlies the types of responses that turn off T cells, that's where cancer is really kind of focused in and really been able to show the rest of us, how do you modulate that potential interaction? So I think that's where really the focus, or at least us, would kind of come into play is as we're designing vaccines to try to generate T cells for infectious disease, can we also bring in some of these T cell modulatory molecules that have really been effective in cancer?
So I'm going to ask you both for a very brief response to my final question, which is, what's your biggest barrier? Georgia?
Well, I say that the biggest barrier sometimes is having the resources to move quickly on a particular problem. And I think it gets back to when we think about how we even how we invest or we hire in people, we invest in the people, right? And not necessarily the project. And if we lead with that, if we lead with the idea we're investing in a person, they're going to pick the best projects and have the fastest endpoint to whatever the goal that they're trying to achieve. So I would say that's probably the biggest barrier. We just need to go ahead and invest in the people first.
In fact, as you know, that's the fundamental idea around the Duke Science and Technology Initiative is we have to keep investing in the people and the talent and the areas of science that we're kind of anticipating are going to be necessary for the next challenge. And also the infrastructure and all the other pieces that go along with it. Kevin, what's your biggest barrier?
Wow. I think that's a hard question for me to answer, actually. If I was to look at it, I would say probably time is always the biggest challenge for me.
Yeah, that's true.
So there's a lot of different ways that we are trying to tackle different pathogens. So between, working on HIV and working on SARS-CoV-2, I think time usually is probably the biggest thing. So timing can be, being able to focus enough attention on a certain pathogen to move a study all the way to completion. Sometimes it means being able to, get the right documentation in place so that we can do a particular experiment, and we need to do it at a certain rate before, either the vaccine that we're making is moving forward to the next stage or something like that, where we're up against a, kind of a deadline. And so, time tends to be the thing that keeps me up at night. It's just trying to make sure that we are on time and that we're going to meet the milestones, and that if there is some, roadblock where we need to get things in order, that we have those done and the projects can move forward at the rate they're supposed to.
Well, certainly the time element played out during the pandemic. I don't think any of us have felt the pressure that we felt to come up with diagnostics, therapeutics, vaccines in a time frame that never experienced before. But I want to end by, first of all, thanking both of you. You are extraordinary, both as individual scientists as well as the teams that you've built. I am so proud and so appreciative of the work that you do. With that, I'm going to turn it over to Bree to close us out. Thank you.
Thank you, Dean Klotman. And to our wonderful speakers, Dr. Tomaras and Dr. Saunders, for a great discussion, and to our audience for joining us.